by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. DNA replication is one of the most basic processes that occurs within a cell. Each time a cell divides, the two resulting daughter cells must contain exactly the same genetic information or DNA as the parent cell. To accomplish this, each strand of existing DNA acts as a template for replication. DNA replicated. Replication occurs in three major steps. First, the opening of the double helix and the separation of DNA strands. Second, the priming of the template strand. And lastly, the assembly of the new DNA segment. During separation, the two strands of DNA double helix uncoil at a specific location called the origin. Several enzymes and proteins work together to prepare or prime the strands for duplication. Finally, a special enzyme called the DNA polymerase organizes the assembly of the new DNA strands. The following description of this three-stage process applies generally to all cells, but specific variations within the process may occur depending on organism. DNA is the genetic material that defines every cell. Before a cell duplicates and is divided into new daughter cells through either mitosis or meiosis, biomolecules and organelles must be copied to be distributed among the cells. DNA found within the nucleus must be replicated in order to ensure that each new cell receives the correct number of chromosomes. The process of DNA duplication is called DNA replication. Replication shows several steps that involve multiple proteins called replication enzymes and RNA. In eukaryotic cells such as animal cells and plant cells, DNA replication occurs in the S phase of interphase. During the cell cycle, the process of DNA replication is vital for cell growth, repair, and reproduction in replication. Step 1. Replication fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into two single strands. DNA has four bases called A. Adenine, T. Thymine, C. Cytosine, and G. Guanine that form pairs between the two strands. Adenine only pairs with thymine or A to T, and cytosine only binds with guanine, letter C to letter G. In order to unwind DNA, these interactions between base pairs must be performed by an enzyme called the DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into Y shape known as the replication fork. This area will be the template for replication to begin. DNA is directional in both strands, signified by a 5 and 3 end. This notation signifies which side group is attached to the DNA backbone. The 5 end has a phosphate group attached, while the 3 end has a hydroxyl group attached. This directionality is important for replication as it only progresses in the 5 to 3 direction. However, the replication fork is bidirectional. One strand is oriented in the 3 to 5 direction, which is the leading strand, while the other is oriented 5 to 3, which is the lagging strand. The two sides are therefore replicated with two different processes to accommodate the directional difference. Where the replication begins. Primer bonding. The leading strand is the simplest to replicate. Once the DNA strands have been separated, a short piece of RNA called a primer binds to the 3 end of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzyme DNA elongation. Enzymes known as DNA polymerases are responsible for creating the new strand by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria, polymerase 3 is the main replication enzyme, while polymerase 1, 2, 4, and 5 are responsible for error checking and repair. DNA polymerase third binds to the strand at the side of the primer and begins adding new base pairs complementary to the strand during replication. In eukaryotic cells, polymerases alpha, delta, and epsilon are the primary polymerases involved in DNA replication. Because DNA replication proceeds in the 5 to 3 direction on the leading strand, the newly formed strand is continuous. The lagging strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase then adds pieces of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjoint. Step 4. Termination. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all the RNA primers from the original strands. These primers are then replaced with appropriate bases. Another exonuclease proofreads the newly formed DNA to check, remove, and replace any errors. Another enzyme called DNA ligase join Okazaki fragments together forming a single unified strand. The ends of the linear DNA present a problem as DNA polymerase can only add nucleotides in 5 to 3 direction. The ends of the parent strands consist of repeated DNA sequences called telomeres. Telomeres act as a protective caps at the end of chromosomes to prevent nearby chromosomes from fusing. A special type of DNA polymerase enzyme called telomerase catalyzes the synthesis of telomere sequences at the end of the DNA. Once completed, the parent strand and its complementary DNA strand coils into the familiar double helix shape. In the end, replication produces 2 DNA molecules, each with one strand from the parent molecule and one mutation would not occur without enzymes that catalyze various steps in the process. Enzymes that participate in the eukaryotic DNA replication process include 1. The DNA helicase, which unwinds and separates double-stranded DNA as it moves along the DNA. It forms the replication fork by breaking hydrogen bonds between nucleotide primase, a type of RNA polymerase that generates RNA primers. Primers are short RNA molecules that act as a template for the starting point of DNA replication. Third is the DNA polymerases. These are synthesized new DNA molecules by adding nucleotides to leading and lagging DNA strands. Fourth is the topoisomerase or the DNA gyrase. Unwinds and rewinds DNA strands to prevent the DNA from becoming tangled or supercoiled. Is the exonucleases group of enzymes that remove nucleotide bases from the end of a DNA chain. Next is the DNA ligase. It joins DNA fragments together by forming phosphodiester bands between nucleus replication tape. In the prokaryotic bacterium, replication can occur at a rate of 1,000 nucleotides per second. In comparison, eukaryotic human DNA replicates at a rate of 50 nucleotides per second. In both cases, replication occurs so quickly because multiple polymerases can synthesize 2 new strands at the same time by using each unwound strand from the original DNA double helix as a template. One of these original strands is called the leading strand, whereas the other is called the lagging strand. The leading strand is synthesized continuously. In contrast, the lagging strand is synthesized in small separate fragments that are eventually joined together to form a complete newly copied strand.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
When DNA replicates, its strands are separated by the enzyme helicase. Single-stranded DNA binding proteins keep the strands from re-annealing. One DNA strand encodes the leading strand, which forms from its 5' to its 3' end, using DNA polymerase III. No problem here, but the lagging strand presents problems. It has to form from 5' to 3' too. It forms in pieces called Okazaki fragments. First, an RNA primase lays down an RNA primer. Then, DNA polymerase III lays down new DNA. The process repeats again and again. DNA polymerase I replaces the RNA primers with DNA. Finally, DNA ligase links the Okazaki fragments.